Hello, welcome to Care ALS Plus Capsule Episode 3. Today we've got some really interesting stuff about Alzheimer's disease.
AD.
AD, exactly. So we're going to kind of dive into what we can learn about it from this, what might cause it, what it does in the brain, how scientists are trying to track it, and maybe some of the ways doctors are thinking about treating it. We'll try to boil it down to the important stuff.
Sounds good.
So to start us off, Alzheimer's, it's a brain disease, right? Millions of people around the world have it. And it's basically where people start having trouble with their memory and thinking. And as people get older, it becomes more common.
Yeah, that's the tough part about it. We have some therapies for dealing with symptoms, but we're still really trying to figure out what's going on at the basic level in the brain so we can come up with better treatments.
Okay, so let's get into that. In the brains of people with AD, what are some of the things scientists see? I've heard about these things called tangles and plaques.
Right, those are the two hallmark pathological signs that we see in AD brains.
Okay, can we break those down? Tangles, what are those?
So tangles, you can think about them as like knotted up threads inside the brain cells, the neurons. These threads are made up of a protein called tau. And normally, tau helps to keep things organized inside the nerve cells. But in AD, it sort of changes shape and starts clumping together, forming these tangles.
So it's like the inside of the cell is getting all messed up.
Yeah, you can think about it like the support system of the brain cells is collapsing.
That makes sense.
Yeah.
And what about the plaques? Where do those fit in?
Plaques are a bit different. They're these sort of sticky deposits that build up outside of the brain cells.
Oh, so outside the cells this time.
Exactly. And they're made up of a protein fragment called amyloid beta, or A. This A comes from a larger protein. And in AD, these fragments start to stick together and form these plaques.
So it's like the brain is getting clogged up, both inside and outside the cells.
In a way, yeah. And your material also mentions things like oxidative stress, which you can think about as sort of like rust forming in the brain. And then there's also the imbalance of certain metals, like iron and copper and zinc, which can also cause problems for brain cells.
It's a whole bunch of stuff going wrong.
Yeah, unfortunately.
Now, I know there are different types of AD. Can you walk us through those?
Sure. So the most common type is what we call late onset AD, and that usually starts after the age of 65. But there's also early onset AD, which is less common and can start much earlier, sometimes even between the ages of 30 and 65. And then we also talk about whether it runs in families. If it does, we call it familial AD, and that's usually caused by specific gene changes that are passed down through families. But most cases are what we call sporadic AD, and that means there's no clear family connection.
So genes are involved, but not always in a straightforward way. What about a person's overall health? Does that play a role in AD?
Absolutely. Things like high blood pressure, high cholesterol, and diabetes, they can all have a negative impact on brain health. And these kinds of health problems can also make the effects of AD worse.
So it's really important to take care of your overall health. It sounds like these brain changes, these tangles and plaques, they might be happening long before someone even notices anything's wrong.
That's one of the biggest challenges with AD. These changes can begin years, even decades, before someone shows any noticeable symptoms. And that's what we call the preclinical stage. And once someone is actually diagnosed with AD, the typical lifespan after that is about 4 to 8 years. Often, people might first experience what we call mild cognitive impairment, or MCI, sort of like a milder form of memory and thinking problems. And a lot of people with MCI actually go on to develop AD.
So catching it early is crucial.
Exactly. And that's why there's so much focus on trying to detect the disease as early as possible. There is some good news, though.
Oh, good. I could use some good news.
Well, there's some evidence that in some parts of the world, the number of new AD cases is actually going down. And this might be linked to things like better heart health, healthier lifestyles in general, and higher levels of education.
So it suggests that some of the choices we make can affect our risk.
That's the hope.
Okay, so that's a good overview. AD is this brain disease with these tangles and plaques, and it can start silently way before symptoms appear. But let's get into the causes. What actually starts this whole process? Let's talk about the familial type first.
All right, so in familial AD, we actually have a pretty clear picture. It's directly caused by specific changes in genes that are passed down through families. These changes are in genes called APP, PSEN1, and PSEN2.
And those usually lead to that early onset form, right?
Right. Usually it shows up between 30 and 65, and it tends to progress more quickly.
So what do those genes actually do? What are their roles in the body?
Okay, so think of the APC gene as like the instruction manual for making that amyloid precursor protein we talked about.
Right, the one that amyloid beta comes from?
Exactly. So the mutations in this gene can cause the body to make more of that AF fragment, or a stickier type of AF that's more likely to form plaques. And this often happens because the mutations are near where these cutting enzymes in the brain, called secretases, do their work on the APP protein. Now, APP actually has different versions, and the one that's most common in the brain is called APP 695.
So faulty instructions lead to more of the bad stuff.
Yeah, kind of like a misprint in the recipe.
What about PSCN1 and PSCN2, those other genes?
So PSEN1 and PSEN give the instructions for making proteins called presenilins, which are actually part of one of those cutting enzymes, the gamma secretase.
Oh, so they're involved in making the amyloid beta, too.
Right. But changes in these genes usually mean that the gamma secretase ends up making more of the toxic types of AB that clump together easily. Interestingly, these mutations have been found in families with early onset AD, but also sometimes in people with early onset AD who don't have a family history. We call those de novo mutations.
Wow, so even without the family history, those genes can still be a problem. Okay, so that's the familial type. But what of the sporadic kind? Most people with AD have that type, right?
That's right. Sporadic AD is much more common, but also more complicated. We don't see those same inherited gene changes. It seems to be a mix of your genes, things in your environment, and other health issues you might have. The biggest genetic risk factor we've found for late onset AD is a gene called APOE, and it's thought to be involved in like 99% of these cases.
APOE, I think I've heard of that. What does it do?
APOE is the instruction manual for a protein that helps to move cholesterol around in the brain, and it's mainly made by these cells called astrocytes. Now, there are three main versions of this APOE gene: A2, A3, and BOR4.
And the A4 version is the one that's linked to a higher risk, right?
Exactly.
So how does it do that? How does APOE increase the risk of getting AD?
Well, we're still figuring out all the details, but we know that APOE affects amyloid beta in a few different ways. It influences how A clumps together and how it gets cleared away from the brain. Now, here's the interesting part. The different APOE versions have different effects on this clearance. A2 is the best, then A3 and A4 is the worst. So having the APOE version means that AG is not being cleared out as efficiently.
Oh, so it's like it's letting the bad stuff build up.
Kind of. And to make matters worse, APOE4 can also bind to A, forming those plaques, and it seems to help turn the individual A molecules, which aren't as harmful on their own, into those more toxic clumps.
So it's like APOE4 is helping the sticky stuff stay sticky. What about those environmental factors you mentioned? What kind of things are we talking about?
Well, the research is still ongoing, but some potential culprits include things like pesticides, exposure to certain metals, and some industrial chemicals. The thinking is that these things might damage brain cells or mess with normal brain processes, making someone more susceptible to AD.
So it's not just our genes, it's also the things we're exposed to in our environment that can play a role. Now, I know a lot of the focus has been on amyloid beta and this idea that it's the main problem, but are there other theories about what causes AD?
Oh, definitely. The amyloid theory has been very important, but there are other ideas out there that are also important. One of the earliest ones is called the cholinergic hypothesis. And this one focuses on the loss of brain cells that use a chemical messenger called acetylcholine.
Acetylcholine, that's important for memory, right?
Exactly. It's crucial for memory and learning. So damage to these cells means there's less acetylcholine in important brain areas, leading to those cognitive problems we see in AD.
So it's like a communication breakdown in the brain.
That's a good way to put it. Another big idea is the neuroinflammation hypothesis. This one suggests that a long-term inflammatory response in the brain involving these immune cells called microglia and astrocytes can actually contribute to the damage.
Inflammation, isn't that usually a good thing? Like when you get a cut, it helps it heal.
Usually, yes. But if it goes on for too long, it can actually start to harm cells. And interestingly, that oxidative stress we talked about earlier, that brain rust, can actually trigger this inflammation.
So it's like the brain's defense system is going haywire and causing more harm than good.
That's the idea.
You mentioned oxidative stress earlier. Could that also be a cause on its own?
Oxidative stress is definitely a major player in AD. The idea here is that an imbalance between damaging substances, those reactive oxygen species, and the brain's natural defenses, the antioxidants, is a big part of the problem. And studies have shown that it can actually increase the production of ADA by messing with those secretase enzymes, making the bad ones work more and the good one work less.
What's a vicious cycle?
Exactly. And to make it even more complicated, the A itself can then increase oxidative stress, like a domino effect.
It feels like all these things are linked together in some way.
They definitely are. And that brings us to another possible cause, metal imbalance. This idea focuses on the abnormal buildup or movement of certain metals in the brain, like iron, copper, and zinc. For example, too much iron can lead to more of that brain rust and can also increase the problems with Al and Tau.
So it's like the brain's delicate chemical balance is being thrown off.
Right. And there are even more potential causes being investigated. For example, there's some fascinating research on the connection between the gut and the brain.
The gut, what does the gut have to do with the brain?
Well, the idea is that an imbalance in the bacteria that live in our gut, what we call gut dysbiosis, can lead to inflammation throughout the body. And this inflammation can then affect the brain and potentially worsen AD.
Wow, the gut influencing the brain in Alzheimer's. That's amazing.
It is. And then there's the brain's own cleaning system, which also seems to play a role. This is called autophagy. And it's basically the process where cells get rid of waste products, including those A and tau proteins. But if this system isn't working properly, then those harmful proteins can build up.
So it's like the garbage disposal system is broken.
Exactly.
It sounds like there are so many different angles to this. Is there one single main cause, or is it more complicated than that?
Well, that's the million-dollar question. Even with all the research that's been done, we still don't have a single universally agreed-upon cause for Alzheimer's disease. It seems to be a complex interplay of many factors, all contributing in different ways.
Like a puzzle with many pieces.
Exactly.
So we've talked about what might cause it. Now let's get into how it actually plays out in the brain. What's the pathophysiology of AD?
So the exact sequence of events is still being researched, but the main things we see are the buildup of those ABA plaques and tau tangles, the loss of brain cells, and the dysfunction of synapses, those connections between brain cells.
Right, and as we've discussed, inflammation, oxidative stress, problems with energy production in brain cells, and metal imbalances are all part of this process.
They are.
Let's focus on amyloid beta for a minute. How does it go from being this normal protein fragment to these harmful plaques?
Well, as we said, A is produced when the APP protein is cut by those secretase enzymes. Beta secretase starts the cut, and then gamma secretase finishes it, releasing the A fragment. There's also another enzyme, alpha secretase, that can cut APP, but it does it in a different place and doesn't produce A. Now, the A42 form, that's the one that's particularly prone to clumping together. And as we discussed, genetic changes can increase the production of A, and risk factors like APOE4 can affect how quickly it gets cleared away. Now, what's interesting is that the individual AP molecules might actually have some normal functions in the brain.
Oh, so they're not all bad.
Right. But it's when they start to form these small soluble clumps called oligomers that they become really toxic to brain cells. The main idea, what we call the amyloid cascade hypothesis, suggests that this buildup of A is the first domino that falls, leading to the problems with tau and eventually the loss of brain cells. But this hypothesis is still being debated.
So the key takeaway here isn't just about the plaques themselves, but also these smaller, more soluble forms of A that are causing trouble. Okay, now what about tau? How does that become a problem?
So tau, remember, it's that protein that helps stabilize the internal structure of brain cells. But in AD, it gets these extra phosphate groups attached, and we call this hyperphosphorylation, and this causes it to detach from the microtubules and form those tangled threads inside the neurons. And these tangles are thought to disrupt the normal functioning of the brain cells. Interestingly, tau problems often start in the areas of the brain that are involved in memory.
Which makes sense, given that memory problems are one of the first signs of AD.
Exactly. And it seems like the spread of these tau tangles to other parts of the brain is linked to the presence of those A plaques. It's like the Akins setting off a chain reaction.
One problem leading to another. The materials also mention something called synaptic dysfunction. Why is that important in AD?
That's a really important point. There's growing understanding that AD is fundamentally a disease of the synapses, those connections between brain cells where all the communication happens. And both AS and tau are thought to damage these connections, making it harder for brain cells to talk to each other.
So it's like the communication lines are getting cut?
In a way, yes.
Hmm.
And to add another layer of complexity, the brain has its own waste removal system that involves these structures called endosomes and lysosomes.
Like a cellular recycling center.
Exactly. And these are really important for keeping the synapses healthy. But in AD, these systems can become dysfunctional, which allows AS and tau to build up even more. And as we mentioned before, genes like presenilin-1 and APOA4 can influence how well these systems work.
So it's like the trash isn't getting taken out, which makes the whole problem worse.
That's a good analogy.
Okay, let's shift gears a bit and talk about something a bit more hopeful. Biomarkers. What are these, and how are they helping us understand and manage AD?
Biomarkers are like biological clues that can tell us what's happening in the body. And in AD, we're looking for biomarkers that can help us diagnose the disease earlier and track how it's progressing. We can find these biomarkers in different places, like the cerebrospinal fluid, which is the fluid that surrounds the brain and spinal cord, and also in blood tests.
So we can actually measure these things and get a glimpse into what's happening in the brain.
Exactly. And we could also use brain imaging to see some of the changes associated with AD.
Okay, let's break this down a bit. Let's start with those cerebrospinal fluid biomarkers, or CSF biomarkers. What kind of information can we get from those?
In the CSF, we often see lower levels of A42. That's because it's getting trapped in those plaques in the brain. We also see higher levels of total tau, and a specific type of tau called phosphorylated tau, which is a sign that there's damage to brain cells and that those tangles are forming. And the ratio of A42 to another form of a fat called A40 in the CSF, that can also be helpful in making a diagnosis. And researchers are also looking at other things in the CSF that might indicate inflammation or other processes that are involved in AD.
So CSF analysis can give us some pretty direct insights into what's happening in the brain. What about imaging? What kind of brain scans are helpful in AD?
Brain scans can be really useful too. We have something called amyloid PE scans that can actually show us those amyloid plaques in the brain. There are these special tracers that are used in the scans that light up the plaques, and there are also tau PE scans that can do the same thing for those neurofibrillary tangles. Interestingly, tau P seems to be even better than amyloid PE at predicting cognitive decline. We also use another type of scan called FDG-PE, which measures how much glucose the brain is using. And in AD, we often see reduced glucose use in specific areas of the brain. And then there's the standard MRI, which can show us things like brain shrinkage, but it's not as specific for AD as the other types of scans.
So imaging gives us a visual picture of what's happening in the brain. What about blood tests? Are those helpful in diagnosing AD?
Blood tests are a very active area of research right now. While they're not quite as accurate as the CSF tests yet, we're making a lot of progress. Researchers are looking at things like the ratio of A42 to A40 in the blood, as well as those phosphorylated tau proteins. There's even a blood test for the A42-A40 ratio that's been approved in some places. The goal is to have simple blood tests that can be used to screen people for AD, although they might still need to be followed up with more specific tests. And scientists have also come up with a way to classify AD based on these amyloid, tau, and neurodegeneration biomarkers. It's called the ATN framework. And the idea is that combining multiple biomarkers, maybe even in a single blood test, could give us a more complete picture of what's going on.
That would be amazing to have a simple blood test that could help us detect AD early. But it sounds like there are still some challenges to overcome before we get there.
Oh, for sure. It's not easy, but it's a really promising area of research.
Now, let's talk about the treatments. What are the current options for people who have been diagnosed with AD?
So right now, the drugs that are approved by the FDA mainly focus on managing the symptoms of AD. We have what are called cholinesterase inhibitors like donepezil, and these work by increasing the levels of that important chemical messenger, acetylcholine, in the brain.
Right, because we talked about how acetylcholine is important for memory and learning.
Exactly. So boosting its levels can help improve memory and thinking in people with mild to moderate AD. And there are different ways to take these medications, like pills and skin patches. We also have a drug called memantine, which works on a different brain chemical called glutamate. And memantine is usually used for people with moderate to severe AD to help with their thinking, daily activities, and behavior. But it's really important to understand that while these drugs can help with symptoms, they don't actually stop the disease itself from progressing.
So they're more about improving quality of life, not actually curing the disease.
That's right.
What about treatments that actually try to modify the disease, to slow it down or even stop it?
That's where things get a lot more challenging and where a ton of research is being done. There's a drug called sodium olemanate that has been conditionally approved in China. And it's thought to work by reducing inflammation in the brain that's linked to problems in the gut and by interfering with the clumping of abaya. Then we have these monoclonal antibodies that target eye, like aducanumab, lecanemab, and donanemab. And these have shown some promise in clearing those plaques from the brain and slowing down cognitive decline in clinical trials.
So they're actually targeting the underlying biology of the disease.
They are, which is really exciting. But these treatments are not without risks. Some people experience side effects like brain swelling or bleeding, and they're also very expensive. And the long-term benefits are still being studied.
So it's a step in the right direction, but we still have a ways to go.
Yes, definitely.
It sounds like targeting amyloid is still a major focus in AD research, but are there other approaches being explored?
Absolutely. Researchers are looking at lots of other potential targets. Some are trying to block those secretase enzymes that produce DAVR in the first place. Others are trying to boost the activity of the alpha secretase, the one that prevents AR from forming. And then there are efforts to develop drugs that prevent AR from clumping together, or vaccines that could help the body clear it away. Tau targeting therapies are also a big area of research, trying to prevent tau from tangling or even clear away the tangles that have already formed. And given the role of inflammation in AD, there's a lot of work being done on anti-inflammatory drugs and drugs that can modify the activity of those immune cells in the brain. And there are also studies on antioxidants to combat that brain rust, drugs that can address those metal imbalances, and therapies that target the energy-producing parts of brain cells. It's a really dynamic and complex field of research.
It sounds like there's no single magic bullet for AD. It's going to take a multi-pronged approach to really tackle this disease.
That's exactly right.
Well, we've covered a lot of ground today. We've talked about the potential causes of Alzheimer's, how it affects the brain, the tools that scientists are using to study it, and the different ways they're trying to develop effective treatments.
What I find really fascinating is just how many different things might be going wrong in the brain in Alzheimer's disease. You've got the buildup of these specific proteins, you've got problems with communication between brain cells, and you've even got the body's own immune system playing a role. It really highlights the complexity of this disease.
And it's clear that early detection and finding ways to intervene early in the disease process are becoming increasingly important. And the advances we're seeing in biomarker technology are crucial in that effort.
They really are. It's an exciting time to be in this field, even with the challenges we face. The research is constantly moving forward, with new clinical trials and a deeper understanding of the disease emerging all the time. And I'm hopeful that this will eventually lead to more effective treatments for the millions of people affected by AD.
And it's important to remember that while we've been focusing on the science today, Alzheimer's disease has a huge impact on the lives of individuals, their families, and those who care for them. Providing support and care is just as important as finding new treatments.
It absolutely is. Thank you for taking the time to explore this important topic with me today.
Thank you for joining us.
We'll see you next time.